And it is actually a great pleasure to introduce today Dr. Aaron Harmon, the Hardy Fellow, which is a very prestigious fellowship in conservation biology that comes with a couple stipulations here, one of which is to teach a course in conservation biology for one of the concentrations here at Harvard, the ESPP, or Environmental Science and Public Policy. It's a very important course that introduces a lot of people to conservation biology, and there's no one better than Aaron for doing that type of work, as his students actually tested this year. He got a 5.0 out of 5.0 evaluation. He did a lot of new innovative things in his class, among other things because Aaron has always been very interested in pedagogy. He's one of the few scientists who publishes real scientific papers related to his research, but also papers on how to make science more available and in definitive in pedagogy. Dr. Harmon was an undergrad student at University of Vermont, from not too far from here, but then he decided to move to the West Coast of the U.S. He really liked it there, so did his master's and PhD at the Scripps Institution of Oceanography, which is one of the main places that people gain trained in marine biology in this country, in San Diego. But then he decided to go back to the East Coast, to the Smithsonian Institution at the Natural History Museum, in National Museum of Natural History in Washington, D.C., where he worked with some of the most creative people in conservation biology and started some of the research that he's going to be talking about today. After being at the Smithsonian, he obtained the Hardy Fellowship, and he's been with us now since January, although he was already living in Cambridge, and we got to see him around now and then. His fellowship has actually been extended for another 6 months, in part because we wanted him to teach again his very successful course. So he's the first Hardy Fellow who's actually going to be a Hardy Fellow for a year and a half, and that is really good for us, and I hope that for Aaron as well. Aaron has been at the forefront of conservation biology for quite some time now, introducing a lot of very innovative science into what he does. And he has authored or co-authored almost 20 scientific papers in many of the big journals in the field, like the Proceedings of the National Academy of Sciences or Current Biology, Coral Reefs, and also has had a couple pieces of opinion in the weekly journals that we all want to publish in, like Nature or Science. His work spans many areas of marine biology and marine ecology, and especially conservation biology, and anthropogenic effects on the marine ecosystems, and especially focusing on invertebrates, and his favorites obviously are corals. So with this, let's hear him and see what he has to tell us. And let's welcome Dr. Aaron Harmon, please.
Thanks. All right. Well, thank you, everyone, for being here tonight. And thank you, Jane and Gonzalo, for those wonderful introductions. I'm extremely excited to be here tonight to talk to you primarily about my work studying corals. And what I study in particular is early life stages of corals, thinking about how parents provide things to their offspring and what implications that has for how well their offspring do when they go out into the world. And I will start by saying that the early life stages of corals are particularly perilous. There's a lot of mortality, but there's also a huge variety of traits in coral larvae. And some of those traits may actually help these animals to survive into the future in a rapidly changing environment. And I should point out here that I'm actually showing this progression in these photos. What you're seeing here is a broadcast spawning coral. Each circle there is an individual polyp, an individual coral, and what it's doing is it's bringing a bundle of egg and sperm into its mouth. It's going to release that in synchrony up into the water column. Hopefully it will be fertilized. This is under microscope looking at the eggs after fertilization, starting to get cell cleavage, and then that is a settled larva that has now started to make its calcium carbonate skeleton. So I'm going to start the story today with kind of what we consider some of the most modern, or the beginning of modern coral reef ecology in the mid to late 1800s, with two very prominent scientists of the time, Alexander Agassiz and Charles Darwin, and their search for the origin of coral reefs. Alexander Agassiz, of course, was one of the founders of the MCZ, and so that hits really close to home. And both he and Darwin were captivated by coral reef ecosystems. And part of the captivation came from the fact that coral reefs just seemed to spring out of nowhere in what they called a marine desert. That is, they came up out of nowhere in the ocean where it seemed like there wasn't a lot of food around us. They wanted to understand why that was the case. They were also just generally amazed by coral reef biodiversity. That is, the diversity of organisms they saw in coral reefs, the diversity of structures they formed, and the diversity, generally speaking, of life that they saw on coral reefs. And so at the time, they lacked a lot of the tools to really dig down deep into this biodiversity. But to that, I would say, if they only knew. So what we have found since the time of Agassiz and Darwin is that about 25% of species that live in the ocean live on a coral reef. It's estimated that about half of the species that live in the ocean live on a coral reef at some point in their life, so maybe early or late in their life. And perhaps more staggering is that there are an estimated 36 animal phyla on Earth, and we believe that 32 of them are found on coral reefs. In contrast to that, rainforests, which are considered the biodiversity analog of coral reefs on land, only have 9 of these 36 animal phyla. So this gives you a sense of just how biodiverse coral reefs are, and that doesn't even begin to think about all the non-animals, bacteria, fungi, archaea, all sorts of other things that also have an immense degree of biodiversity. But as many of you have probably heard, and what I think might have motivated many of you to come tonight, is that coral reefs appear to be on the brink of collapse. These are 2 scientific papers I'm highlighting. These were early 2000s, so we're talking 15 years ago, people were noting the decline. If you search the literature for coral reef decline, there's 1,700 papers. 1,600 of them have come since 2000. And then if you look in the popular media, the concept of coral reef decline is popping up more and more. In particular, just a couple of weeks ago, the IPCC report projected that within the next 20 to 30 years, if there aren't significant changes, we could lose anywhere from 70% to 99% of the remaining coral reefs, which is already a very depleted number of reefs relative to what we had in the past. And so to start kind of with some of the sad stories, if we think about everything that we're losing when we lose coral reefs, I'm going to get sad and then try to build us back up. We're losing industry and we're losing jobs. It's estimated that coral reefs have a value of somewhere around $1 trillion in terms of industries and jobs that they're contributing. And that can be anywhere from small scale to large scale fisheries, as well as tourism, diving, people traveling to go see coral reefs. Coral reefs are also incredibly important sources of food and nutrients. They provide one of the primary, if not only, sources of protein to many people in often impoverished countries where it's more challenging to get protein. And they're also very important sources of nutrients. I was reading recently things like zinc, vitamin A, deficiencies are expected to rise for considerable amounts of the human population. Somewhere in the range of 10 to 20% of the human population depend on fish for those vital nutrients, and they're going to no longer have access to them as fisheries are declining. We're also losing shoreline protection, which, of course, is incredibly important. If anyone has paid attention to what's going on with hurricanes recently, coral reefs build these massive structures, and they can actually protect shorelines. And this picture over here is actually on land, but what this cliff I'm showing you here is all coral. So it highlights that corals not only protect shorelines underwater, but old corals on islands that have risen up actually contribute to the building of islands. So many islands are actually literally built on corals. And then finally, for those of you who may not be interested in ever traveling to a coral reef, I'll point out that we're losing medical potential. We're losing a lot of potential to find new molecules that can be turned into medicine. Just to highlight a couple, palytoxin was one that was turned into a cancer-fighting drug, and another one more recently were proteins that are used to fight HIV. And to highlight some of my own work, I did a project, my previous project with the Natural History Museum at the Smithsonian was collecting these things. So these are ARMs, or autonomous reef monitoring structures. We put them out on reefs, leave them for a few years. We estimate around 70 to 80% of the life on the reef recruits to it, and then we collect it and see. And so what I did is I looked at all of the molecules, what we call small molecules, generally in the area of lipids, that are particularly interesting because they confer functions. Some of these functions may be interesting in medicine. And what we found is we have a vast unknown molecular diversity. So only 0.6%, 62% of the roughly 9,000 molecules we could detect, we were able to figure out what they were. So those only 0.62% were actually known to science. And these were from reefs in the most biodiverse part of the world, at least as far as the ocean is concerned, what we call the Coral Triangle, Indonesia, Philippines, and other parts of the world. And so we found that there was just this incredibly small number of molecules that we could identify. And what I'm showing you here, every single one of these little circles is a different molecule. The ones on the far left are linked to other molecules because they're structurally similar to one another. And all these non-linked ones didn't even have any structural similarities to any other molecules in the whole data set. And I've color-coded them by whether they were found in multiple areas, gray, or just found in one area, a color. And you can see a vast number of these molecules are only found in one area. So it shows you that all this molecular novelty is actually distributed across a wide number of regions. And so we're beginning to try to understand what some of this molecular diversity means from coral reefs. And so what I'm doing, what I've done recently, is to look at how these molecules are related. Say we're related to a drug that we already know, but this is sort of like searching for a needle in a haystack. And all of this is to say that we as scientists aren't going to be able to keep up in terms of how rapidly we're figuring out what these molecules do and relating them back before we lose coral reefs entirely. So what is causing this decline in coral reefs? I'm going to tell this story sort of in three parts. I've admittedly oversimplified the ecosystem, but I think it's an easier way to understand it. So at the base, we have corals. Corals are a cnidarian. They're related to anemones and jellyfish, and they're an extremely efficient recycling system. Really, all of a coral reef is an extremely efficient recycling system. Corals harbor within them single-celled algae, which are called dinoflagellates. And these algae photosynthesize just like a plant, but what they do is they release these products of photosynthesis, lipids, sugars, things like that, to the coral. The coral then consumes it. The waste of the coral in the form of nitrates and phosphates goes back to the algae, which it then uses for photosynthesis. The algae in return gets protection, and the coral gets food. Somewhere in the range of 70 to 80 to 90 to even 100% of its food comes from these algae. And I should point out here, too, that these algae are not just one type. There's a whole range of types that are acclimated to different sorts of environments. And why that's important will come up a bit later in the talk. So here we have a single coral individual, or a polyp, like what I showed you in my introductory slide. What this polyp does is it builds carbon copies of itself, essentially. And it's all embedded in a skeleton. And so the corals build out a colony. Excuse me. And so while this polyp may be about 1 millimeter in diameter, polyps together can grow this coral colony. This coral colony could easily, this is actually a life representation of it, this could easily be as tall as I am, or even significantly taller than I am. And so in aggregate, corals form the base of the ecosystem that coral reefs are built on. On top of that, adding another layer, we have what we call the lawnmowers. So the herbivores, things like fishes and sea urchins. And why they're particularly important to this story and to the coral story is that coral are competing with algae on the reef. And these are not the same type of algae that they have within them. These are things that look like a plant in your garden or look like your lawn. And so they're competing with corals for space on the reef. What these lawnmowers are doing, as you might imagine, is they're going around and mowing the lawn. They're keeping these algae at bay. They're taking that algal biomass, putting it into themselves, and building up biomass in fish and urchins instead of in algae. On top of that, we have the scary stuff, or the stuff that people want to go dive and see. We have things like top predators, sharks, and other large fishes that are eating the smaller stuff. And so on a really healthy reef, you actually have most of the biomass get sucked up into these top levels. And so as a result of that, you don't have much at the bottom, you have a lot at the top, and you have extremely efficient recycling going on a coral reef. Well, so what we've done is reversed this recycling process. So we've removed the top predators first, and then we've also removed the lawnmowers by overfishing them, taking out too many, eating too many. And as a result of that, these algae that I was talking about start to get a foothold. So you see some turf algae here and some macroalgae spotting up here because there's no longer things there eating them away. We've also fertilized the reef, so literally with runoff of fertilizer from agriculture or golf courses, things like that, but also runoff of human pollution, human sewage and other forms of pollution. And unlike the corals, the algae need to get their nutrients from photosynthesis from external sources. So they're pulling it out of the water. So when we give them a lot of these nutrients, they suck it up, they grow, they start to outcompete corals. And then finally, we've turned up the heat. With climate change, we've increased the temperature such that it's causing corals and algae to disassociate. And so the reason that I've depicted these corals now as white or tan is that if you look at an individual coral, what happens when you turn up the heat is it expels or consumes its symbiotic algae. And the reason for that is at higher temperatures, photosynthesis actually breaks down in the algae and they start harming their coral host, much like a pathogen. And so the coral needs to get rid of it, but obviously it's now lost its primary source of energy. And so if conditions don't return to normal, it's not able to take its algae back up or grow them back up from small populations, that coral will use up all of its energy and die. And so what I'm depicting here is now a dead coral, whereas a few have rebounded. And that is how we go from a relatively healthy reef, like the one on the left, to a relatively unhealthy reef on the right. There are certainly other trajectories to that outcome, but that's sort of the general story. And so I think it's actually pretty staggering to think that in roughly 150 years since the time of Agassiz and the time of Darwin, when we were just trying to figure out how coral reefs as an ecosystem came to be, now 150 years later, we're seemingly on the brink of losing them entirely. So I discussed a little bit about how coral get their energy, and to delve a little bit deeper into it, corals rely on fats, carbs, and proteins just like we do. Their primary source is to get this energy from their algal symbionts, but they can also get it from eating, and they can also get it from stored fats. Stored fats, in particular, the wax esters and the triglycerides, are really important energy sources for corals. Those are going to be really important energy sources throughout the rest of my talk here today. But obviously, when a coral bleaches, now its secondary sources are its primary sources, and it tries to hold on. And so what people have shown is that corals will actually deplete these stored fats over the course after they've bleached in order to try to stay alive. Well, what I'm showing here, which is a single coral polyp devoid of its algal symbionts, is actually relatively analogous to how a coral starts its life, at least if it's starting from an egg or a larva. So the primary source of a new baby coral is stored fat that it got from its parents. So this fat can be anywhere in the range of 60 to 99.9% of its biomass can be these wax esters and triglycerides. And it's burning them up as it's moving around in the water column. Its secondary source can be algal symbionts, but that's only the case for a certain number of species. Only about 29% of coral species pass on symbionts to their offspring. To define some terms here, what I just described of getting the symbionts from the parent is termed vertical transmission. But the other option is to not give the algal symbionts to your offspring and force them essentially to take them up from the environment. And that's about 71% of species that actually have to do that. We call that horizontal acquisition of symbionts. That is, they have to find them in the environment and take them up. And so if we're talking about the way a coral starts, what I was just showing you was a polyp as it would be settled on the reef. But they don't start that way. They start as eggs, they start as larvae that are kind of essentially little blobs of fat moving around in the water column. And there are two primary ways that corals reproduce. The first is brooding, where fertilization happens internally. It happens within the polyp. They then release a larva that's competent to swim. It can swim around. It can settle immediately if it wants to. It moves around in the water column. It then searches the reef for chemical cues that say, hey, this is a good place to live. It settles. It goes through metamorphosis. It starts to build this calcium carbonate skeleton. It restructures all its tissues. In contrast to that, you have broadcast spawning, which is external fertilization, like I showed in that first slide, where corals release these bundles of egg and sperm, or egg or sperm, into the water. Fertilization happens at the sea surface. These now fertilized larvae start to move around. They too then find their way back down to the reef, settle, and go through metamorphosis. And so in these two processes, you go from an adult colony to a larva that's moving around in the water column to an individual that's settled as a single polyp and is going to try to build its own colony. And so what is so special about this process? And when I say what's special about coral larvae, I mean what's unique about them that could potentially help to help these animals persist into the future. And so I'm going to offer a few examples, and then I'm going to focus in on a few. So parental preparation, like I mentioned in the beginning, what coral parents are giving to their offspring. Genetic novelty, the fact that we're getting genetic recombination when fertilization happens. Mobility, this is a huge one. The fact that coral larvae can move. Adult corals, for the most part, cannot move at all. They are affixed to the seafloor. They will not move in their life. They might live for 300 years and they will not move other than to grow outwards. In contrast to that, coral larvae can move. They can go hundreds of kilometers, potentially. The experiences that they have in the water column can also shape how well they do later on and are incredibly important for determining their long-term survival. And then finally, the fact that some larvae are born without symbionts, you have a potential to pick up new symbiont types that may be favorable for the environment that you're living in. And so I'm going to focus on three: parental preparation, larval experiences, and mobility. And so I'm going to give analogs to the three stories I'm going to tell that relate back to humans. And the first is thinking about maternal provisioning of energy. And so what I mean by this is giving fat or any other form of energy to your offspring. And when my wife was pregnant with our son, who's on the left, I was fascinated by how often you heard this question of how much did your baby weigh? How much does your baby weigh? I think new mothers probably got asked this a million times. And I think oftentimes where people have very adequate medical care and babies go through healthy gestation, people are actually thinking about the implications of the pain involved in childbirth when they're asking how much a baby weighed. But I think this question is actually rooted in the fact that we know that baby weight can be a strong indicator of how likely that baby will be to survive and how healthy that baby is when it starts its life. In contrast to that, mothers are also limited in the amount of energy they can give up, right? And so whereas our son was 6 pounds, 11 ounces when he was born, our friend who had 3 healthy triplets, they range in weight from 2.5 to 3 pounds each. And they're big part of the reason for that is that the mom only has so much energy to give. And so we use weight as an indicator of both energy and toughness, that is, health of offspring. And so I've applied that to corals. And before I talk about that, we have to point out the fact that as opposed to humans who generally produce 1 baby at a time, a lot of animals produce a lot more than that. So many species produce many babies simultaneously. And these trade-offs become much more strong when you're producing many, many more offspring. And so my big question was, how does per baby investment in corals influence their survival, influence their success? And so I looked at this in 3 corals, basically to ask, are mothers limited by energy and are all babies the same? And I'm going to focus just in the one on the far left. So the first question I had to ask, of course, is do we see a trade-off in size? What I'm showing on the X here is the number of larvae that a parent released against the size of those larvae. And we actually saw an increase, which is paradoxical to what we would expect. We would expect if you're making more babies, you have to make them smaller. Taking this a step further, we said, okay, maybe it's not just size, but maybe it's the energy content that the babies have. You would think that the babies would need more energy the bigger they get. Well, we found, in fact, that there was no relationship between larval size and the density of the energetic lipids they had. Okay, take that one step further. Now we have a bit more of a smoking gun. What I've plotted, again, is the same x-axis, the larvae released, but against the energetic lipid density. And so what this is saying is that as a parental colony is producing more offspring, the density of energy of each one of them decreases. So for the parent, it seems to be that the parental colony is really constrained by the amount of energetic lipid it has, which makes perfect sense in terms of the importance of that energy. But at the same time, they're still making bigger babies. So what are the implications of that? So I wanted to ask, are larger babies tougher? And do larval experiences matter? That is, the environments they experience as they're moving around in the water column, does that affect them in the long term? So we did this in 2 species, a brooding coral and a broadcast spawning coral. We separated them by size into large and small larvae. We created 1 week in the plankton, which is really just in these falcon tubes, where we put them in either ambient, high temperature, or low salinity conditions. We knew that high temperature and low salinity should be enough to stress them out. We then took them out of there, tested how well they would settle by giving them cues that we know should initiate settlement and metamorphosis. And then we returned them to the reef for about 5 months and looked at how well they survived. And bear in mind, they've gone through metamorphosis, they're now attached. We've put them back on the reef that their parents came from. And so what I'm showing here is the number of settlers that were alive against time. And what is very clear is if you were born large as a larva, you had higher settlement, you had higher survival after you settled than small. And right around this 1 month period, all those that had started out small crashed out to almost nil. Now what if we add on stressful environments and focus just on the large larvae? Well, similarly, if as a larva you'd experienced either high temperature or low salinity conditions, you too crashed out, even though you're back on the reef in relatively ambient conditions, just like where your parents lived, you carry this history of your larval experience. And this to me actually was relatively profound. You have a difference in size at birth, you have 1 week in relatively harsh environments, but I should say it did not initiate large levels of mortality. So it wasn't that they were extremely stressed, but they still carried a signature of it towards the end. And that signature was actually larger after settlement than it was while they were larvae. And so, yes, we find that larger babies are tougher, but no, it does not make them tougher. Larger larvae have higher survival, but it doesn't actually buffer the effects of harsh environments. So it doesn't make them tough if they experience a bunch of harsh environments when they're moving around in the water column. And then secondly, do larval experiences matter? Absolutely. So we saw that a very short larval period had very large implications later on. And so an analogy I like to use for this is smog. Smog, this is a picture of Los Angeles, but smog is an issue in a lot of places. And it's this notion that if you grew up in a place that was smoggy, even just during your childhood, you might go on later, you will go on later to have higher risk of respiratory illnesses and a number of other illnesses that are latent effects of that exposure when you were young. You didn't necessarily get sick when you were young, but you carried it with you and you got sick later in life. So I mentioned that we did this in 2 species, right? And so we did this in this brooder and found that they were sensitive to these harsh environments. But interestingly, in this broadcast spawner, they weren't. They did not experience higher mortality in these harsh environments. We did another study after a very unfortunate oil spill that occurred on Curacao and found that oil contamination had the flipped effect on these 2 species. These bigger brooded larvae were not that affected by oil contamination. Broadcast spawned were very sensitive to it. And so what we generally concluded was that this may be a toughness thing. This may be that bigger larvae have more material inside them and less surface area relatively, and so they're more resilient to things like oil contamination. But it's a different story when it comes to these. So these 2 factors, heat and low salinity, can both influence the symbiotic algae within the coral. And like I mentioned before, these guys had symbiotic algae. Those guys did not. And so we actually hypothesized that it was the presence of the symbiotic algae harming the coral because they were experiencing these harsh conditions. And so that sort of started us on this level of questioning to think about how larvae interact with their symbionts. And so starting with where they acquire them, whether it's parent or ocean, state of the environment, ambient environment, or stressful. Here's what was known in the literature. It was that if you got them from your parent and you lived in an ambient environment, it might benefit you because you get an additional energy source and you could have a longer larval period as a result, more time to search around for a place to live. In contrast to that, if larvae born without symbionts were fed them, they would take them up, but if you expose them to a harsh environment, they would then die. So taking them up and experiencing harsh conditions was detrimental. But there were two gaps, and so we went and we filled in these gaps. So Val Chamberlain worked on the first study with me. She had done fish for her master's, and we convinced her that coral larvae were much cooler. And so she went on to do a PhD in this. And so the follow-up study that she did to ours, which is really elegant, is she took larvae of this species. And what this is really cool about this species is they produce larvae with a huge range of symbiont densities. So some look super pale because they have very few symbionts. Some look very dark brown because they have a lot. So Val separated them into low density, mid density, and high density larvae with symbionts. She exposed them to ambient conditions. Nothing happened. So survival of all of them was very high, regardless of how many symbionts they had. But if she cranked up the heat on them, mortality increased dependent on how dense the symbionts were. So those that had really high density symbionts, larvae with high density symbionts, experienced very high mortality, suggesting to us that even in vertical transmitters, that is larvae that get the symbionts from their parents, if you put them in stressful conditions, they're more likely to die. And that left one more box to fill. So I took the same species, Orbicella faviolata, exposed them to ambient conditions, got them to take up the symbionts. These were actually symbionts we took from their parents and then watched their survival. And it was quite odd. So what I'm showing in the green circles is larvae with symbionts, and the black squares is larvae without, their survival through time. And what we actually found is that if larvae had symbionts, they had slightly higher mortality, which was extremely confusing and sort of the opposite of what we might expect. In this study, we dug a little bit deeper. We looked at the genes they were expressing when they took up the symbionts. And what I'm showing you here, black squares and white squares are non-symbiotic. Circles are the ones with symbionts. Very different responses in gene expression. But there was no difference whether the larvae were exposed to light or whether they were put in constant darkness. Constant darkness meant that their symbionts were not photosynthesizing. So basically what we could show through this and through delving into what some of these genes were is that larvae, when they took up symbionts, were not switching to them as a new energy source. They were also more likely to die, and they weren't decreasing the amount of energy they were using. And so it suggested to us that there were these very fundamental differences between the larva-symbiont in the larva-symbiont relationship if you get them from the ocean versus if you get them from your parents. And you can't just add symbionts to larvae that don't have them and hopes that it's going to boost them up. So what this suggests is that there may be some sort of evolutionary constraints on this relationship. So we delved into this a bit deeper. This, I realize, is potentially scary for some of us, including myself. This is a tree where each one of these circles represents a coral species. This is 232 species. It's not quite all of them, but it's all that we have the following information for, whether they get their symbionts from their parents or whether they get it from the ocean. And what you can see is there's a distribution of those traits across it. And we also can now plot against that whether they reproduce by brooding or broadcast spawning. So why have I combined these two things here? Well, there's an extremely strong association in corals that most brooders acquire symbionts from their parents, most broadcast spawners acquire symbionts from the ocean. And so the reason for this has long been thought to be the following. It's been thought that short dispersing larvae, that is larvae that don't move very far away from their parents, favor taking the same symbionts as their parents. That makes sense, right? They're going to live in the same environment, same type of symbionts benefit them. And that brooding species don't disperse very far. And so brooders give symbionts to their parents or give symbionts to their larvae because they're going to settle close to home. In contrast to that, broadcast spawning is thought to create long distance dispersal of larvae. And long dispersers benefit from being able to acquire symbionts wherever they land, right? So parents don't pass on symbionts because you don't know what type of environment you're going to land in. It might be more advantageous to be able to pick up new symbionts. And I'm going to dub this the dispersal distance hypothesis. There are other iterations of this in other groups as well. But we noticed a few weaknesses with this hypothesis. One is that genetic data has told us that broadcast spawned larvae don't actually disperse as far as we thought they did. So we used to think they would always go very far from their parents, but actually a surprisingly large number settle very close to home. And then secondly, which I think is potentially more concerning, is that larvae don't need to travel long distances to find new environments. Within even a single coral colony that may be this wide, certain symbiont types could be good at the top, certain symbiont types could be good here, certain other ones could be good here. So larvae don't need to travel 100 kilometers to find a new environment that's going to select for a new type of symbiont. And so going back to our studies, what this suggested to us is that instead of what happens after settlement, determining whether it's good or not to have symbionts as a larva, we suggested that instead it has to do with larval experiences. And in particular, the larval experience we focused in on is the high light and temperature environment that arises predictably at the sea surface. Well, which larvae reach the sea surface? It tends to be the broadcast spawners because they're moving from a 3D environment to a 2D plane, which is going to facilitate mixing of egg and sperm. And so the hypothesis then is that if larvae had symbionts and they predictably reach the surface, they're going to be harmed by them, which may be why they don't have them to begin with. And so it brings us to this question of how and why eggs and larvae reach the sea surface. And the reason for that is extremely high concentrations of these lipids called wax esters. Wax esters are a critically important lipid in the ocean, in part because they confer buoyancy, positive buoyancy to things. They help them float, maybe potentially more than any other molecule out there. And then secondly, what helps larvae get away from the sea surface is the fact that they can swim downwards. But the key point here is that a broadcast spawned egg has to be fertilized, it has to go through development, and it can take it half a day to a couple of days until it's able to swim. During that period of time, it's effectively trapped at the sea surface. It's not able to drop down. So this combination of wax esters and not yet being able to swim could potentially trap corals at the sea surface. And so what we did, to put it briefly, is we looked back in the history of corals. And we do this by dating fossils that we have and using molecular techniques. And we looked for past transitions where we thought an ancestor was a brooder and the species today is a broadcast spawner, to look at evolution between these traits. And when we do that, we can actually derive rates. So to say how rapidly or how often did a brooder become a broadcast spawner or a vertical transmitter become a horizontal transmitter. And then from those rates, we can infer selection from the environment. In other words, if a rate is faster, it suggests that it was selected for. And so what I've shown here are these 4 different states that that corals can be in. And I'm going to highlight 2 of them. First is that if you are a horizontal transmitter, that is, you don't pass symbionts on to your offspring, you favor broadcast spawning. You may favor broadcast spawning because it might be favorable because you're mixing gametes. It might be favorable because of other factors, lower inbreeding, things like that. In contrast to that, if you're a vertically transmitting species, there's no selection in either direction. And so what we can infer from that is that there is some cost to vertical transmission if you're a broadcast spawning species. Then to get into this question of whether there's actually selection against larvae spending time at the sea surface, we can look to the wax esters and to the size of larvae. And so our first prediction was that vertical transmitters will produce eggs and larvae with less wax ester. That is, they will be less floaty if they have symbionts. And here's what we found. So this is wax ester content. This is broadcasting species, brooding species. Black circles have symbionts, white do not. What we found is that eggs and larvae with symbionts have lower wax ester content, meaning they're less floaty, meaning they should be able to move away from the sea surface to begin with, or just be negatively buoyant to start. The second prediction we made is that in broadcasters, vertical transmitters produce smaller eggs. Smaller eggs can develop swimming more rapidly, which would allow them to move away from the sea surface. And we find support for this as well. Eggs with symbionts tend to be smaller, meaning they develop more quickly. These larvae can then move away from the sea surface more rapidly. And then finally, which is another, what I think potentially the most compelling piece of evidence, is that if a species evolves brooding from broadcast spawning, you'll see a decrease in wax ester provisioning over evolutionary time. But it will only occur in vertical transmitters. That is, they will become less floaty as a species over time. And so again, I'm showing the amount of wax ester here, and this is the time since a species was a broadcaster. Everything here on 0 is currently a broadcaster. Anything from here, I'm showing you how long ago its last ancestor was a broadcaster, because currently it is a brooder. And so what we find is that in vertically transmitting species, wax ester provisioning decreases through evolutionary time, but only if you're a vertical transmitter. This horizontal transmitter, who's a brooder, is still producing extremely buoyant larvae. And there's only 4 of these species. We're only able to look at 1 so far. But this suggests to us that wax ester content is declining in vertical transmitters. They're becoming less floaty. And so what this highlights is that we had a hypothesis, the dispersal distance hypothesis, that was suggesting processes that occurred after settlement were driving when larvae got their symbionts. But in contrast to that, we're suggesting that actually stress earlier on in life can predict whether or not it's advantageous for species to pass on symbionts to their offspring. And what that suggests, again, is that there's a lot more going on in the early life stages of corals that influence their eventual success. And so this actually brought me to an interesting analog. I've been trying to think about analogs to this story for a bit, and one of them is related to birth timing in humans. So many of you have probably heard that there's a limitation between pelvic size and brain size. That is, humans are born early because we build these big brains and they aren't able to get out. But actually, recent evidence has looked at these predictions and found that they tend to fail upon inspection, and that it may in fact be more related to the energetic limits. That is, as the baby is getting bigger and bigger, requiring more energy from the mother and having high growth rates, and she's just not able to sustain it and needs to give birth to the baby. So this is an example of how what seems intuitively correct to us may actually fall apart a bit on closer inspection. And so this brings up this general question, which gets us back to the question of coral reefs of the future, which is why disperse? So one of the primary reasons for dispersing, that is moving away from your parent colony, is to escape a poor environment. And on evolutionary timescales, it could be actually to move your species, right? So I mentioned coral can only move when they're larvae. Well, if they're going to move, say, further away from the equator to where waters aren't quite as warm, it's going to happen through larval dispersal. Larvae also can be sources in sinks. That is, they can help to reseed areas that maybe have deteriorated. And then on larger scales, it can help maintain genetic diversity. But we don't have a great answer to this question of, can parents control dispersal distance? That is, can they control how far their offspring travel? I have some preliminary evidence that suggests that maybe they do. And so what I'm showing here is wax ester content and the size of larvae. And what I'm showing is that in two species that make positively buoyant larvae, they increase wax ester content with offspring size, presumably making up for the fact that you now have a bigger balloon to float, so you need more of wax esters, which makes you floaty. In contrast to that, the species that produces negatively buoyant larvae makes bigger larvae, but does not add any more wax ester content to them. So it suggests that multiple things are changing at once, wax ester content and size in individual parents, which may allow them to control how far their offspring are dispersing. And so it brings us to this general question of what is going to generate dispersal. And so that's kind of the next question I'm starting to get into in a collaborative group to try to quantify some of these neglected traits and then look at how these species that exhibit these traits are related to one another. And the final piece of this that we're currently working on now in the Giribet lab, in particular with Lihia and Shahan, is we're back to the time of Agassiz. This is Louis Agassiz, actually Alexander Agassiz's father. And the way that we're going back to that time is by using specimens that are here in the museum and trying to get DNA out of them. And why that's helpful is, in theory, we can take the DNA to then look at the structure of populations of the past, to look at how diverse they are, and to look at the genetic diversity and genetic potential of those populations to get at this question of, do the populations of the past look different to the populations of the present? And have the populations of the present started to move poleward? We're particularly fortunate because a lot of the Agassiz samples were taken from Florida. Florida is the fringe of the range of a lot of these species. And so it's a really nice opportunity for us to potentially look back in time at the genetics of corals 165 years ago versus corals today to begin to ask this question of, are corals expanding their range? And then looking at, can we tie that back to some of the traits of the larvae of those species? And so I bring us back to this question of what's special about coral larvae. I focused in on these three factors. And if I'm going to say what's special about each one at the parental preparation level, we found that there's extreme variation in a lot of these larval traits, which could allow for rapid responses to their environment. That is, parents could rapidly tailor, say 1 month to the next, larvae to different environments if there are certain traits that can be advantageous to those environments. But that certain traits, such as vertical versus horizontal transmission of symbionts, are somewhat locked in, and that species possessing those traits may fare better just based on characteristics of their larvae and not characteristics of the adult with environmental change. Found too that larval experiences can last a lifetime. I found in particular that they can be detrimental if you experience harsh environments, and it may explain why certain babies survive to adulthood while others don't when people go out and survey a reef, because we're lacking all of this history of what they experienced as larvae. It also suggests that things such as putting restrictions on environmental impacts during times of broadcast spawning could be particularly important. It also suggests that if we're trying to rear larvae and settle them out in the lab, it's probably not a good idea to stress them out as a way to find the tough ones, because that stress can stick with them, and then once we put them out on the reef, even more of them are less likely to survive. And then finally, mobility, which is one of my favorite topics, if you couldn't tell, to bring up this question of, can corals move to better locations? And if they can, will this actually build the reef to the future, or at least improve the reefs at the fringes of coral ranges as climate change continues to increase? And with that, I'd like to thank a number of people. I highlighted a number of my collaborators, and a lot of them were left off. In particular, I'd like to thank Sarah and Daniel Hurdy for endowing this fellowship, which has been an exceptional opportunity for me. Gonzalo Giribet, who has been an incredible host to me, both personally and scientifically, as well as the lab, which is a group of some of the kindest humans I've ever met, who just welcomed me with open arms, as well as Adam and Jen at the MCZ, in particular in invertebrate zoology, who have been extremely helpful to me as I dig into their shelves and dust off extremely old coral specimens. And so with that, I'd be happy to take any questions.